My name's Ben Lamont. I'm a postdoctoral research fellow in the Drucker Lab. As a postdoctoral fellow, my personal research goals are to further understand the processes of controlling metabolism in order to find pathways that may be useful in treating type 2 diabetes. And the research goals of the Drucker Lab are to understand the biology of the glucagon-like peptides, GLP-1 and GLP-2, and how they can regulate blood glucose. So our combined research goals fit very well together. In type 2 diabetes, the challenge of treating or of controlling blood glucose comes down to the fact that type 2 diabetics don't produce enough insulin from their beta cells to overcome the insulin resistance that they have. There are several therapies now available that approach this problem, but many of them actually have the risk of causing hypoglycemia or, in some cases, weight gain. So one of the advantages of incretin hormones like GLP-1 is that they can lower blood glucose without the risk of hypoglycemia. Okay, so the importance of GLP-1 in blood glucose control. Under normal circumstances, GLP-1 is a peptide that's released from the intestine following meal ingestion, and it has in the body an effect of enhancing insulin secretion from the pancreas. So, based on this idea of it being an incretin hormone, drugs have been developed to target GLP-1 receptors in the pancreas, and these drugs work well in enhancing insulin secretion from the pancreas, but there are also GLP-1 receptors in several other tissues in the body, and GLP-1 appears to have therefore beneficial effects on other tissues. So what we're interested in is finding out more about what the role of GLP-1 receptor signaling in several tissues has. Therefore, our current project actually involves using a novel mouse model to look at the specific role of GLP-1 receptor in the pancreas. Has the human GLP-1 receptor gene introduced into the pancreas, and our results show that in this mouse only the pancreas is the only tissue that is GLP-1 responsive. Without having, and GLP-1 doesn't have any effect in other tissues in this mouse. Furthermore, because this mouse is a mouse that contains a human GLP-1 receptor, we may be able to utilize this mouse to further identify novel GLP-1 receptor agonists, or small molecule agonists that may be used in future treatments for type 2 diabetes. So the next steps in this work, which are being undertaken by our own lab and other labs, is to better understand the precise roles of other tissues in the biology of GLP-1. So us and other labs are developing new models and techniques to tease out the roles of other tissues such as the brain and the nervous system, the heart and the blood vessels, and any other key organs that we identify that contain the GLP-1 receptor.